Good morning, I bid to the lecturers and my fellow classmates. Today, I have a question for you. Are you a robot? If you're not, chances are you have taken drugs or medications before. But how do you know if the medication is the right cure for the disease you're having? That's where transgenesis comes in. Transgenesis is the process of combining DNA or inserting a foreign DNA into a genome using recombinant DNA technology so that the desired property will be transmitted to its offspring. These offspring are called transgenic animals. They provide animal models of human diseases to help researchers find new treatments. And today, me, Liyana, Farah, Najihah, and Najwaa will be discussing on how transgenesis helps in the development of drugs for type 2 diabetes. Type 2 diabetes is a disease where the patient have a high blood sugar due to the impairment of pancreas to produce insulin. Basically, without insulin, the cells in the body cannot use the glucose to produce energy and will stay in the bloodstream. There are two ways to produce transgenic animals, which is through transgene expression and homologous recombination. Transgene expression is the alteration of the gene expression by injecting a foreign gene into the host, whereas homologous recombination is when gene marker knocks out the function of the control gene of an organism, producing animals with a different phenotypes known as knockout animals. This study is carried out to determine which target protein is important for glucose transport, insulin signaling, and secretion. So next, I will pass to Farah.
Thank you, Liyana. So next, I will explain about the methodology that used in this experiment. There are 5 steps that involve. The first one is designing the targeting vector. Second is inserting the targeting vector into the embryonic stem cell. Third is selecting cells. Fourth is injecting cells into the new embryo, and lastly is to isolate and breed the recombinant host cells. The first steps requires to design the targeting vector. The markers NEOR and tyramidine kinase are inserted into the target gene homologous DNA1 and DNA2 to make a targeting vector sequence. Move to the second step, which is inserting the targeting vector into the ES cells. The targeting vector is inserted in ES cell via electroporation, which resulting 2 possibilities, successful cells and unsuccessful cell. The targeting vector in successful cells recombine with target genes and knock out the 1 copy of the target genes. At selecting cell, only the cells that successfully incorporated with the targeting vector into the target gene will survive in the presence of the resistance. Next, the cells that contains targeting vector will be injected into the new embryo, which is the normal developing mouse embryo. Lastly, the final process is to isolate, validate, and breed when the mouse is successfully generated. The chimeric mouse will be bred with a normal black mouse in order to pass down the altered genetic to its offspring. The successful offspring will have a normal version of the knockout gene. So, I will pass to Farah Najihah to explain about the result.
For the result and discussion, there are 3 studies conducted, which is insulin signaling pathway, glucose transport, and insulin secretion. First, insulin signaling pathway. It is discovered when knockout and overexpression insulin receptor, resulting in the death of mice due to hyperglycemia, which is high glucose level. This shows the insulin receptor play a big role in body glucose regulation. Next, there will be deletion of genes IRS1, a protein that integrates signal from the insulin receptor, which leads the mice to resist insulin and stunt their growth. However, IRS1 is not the only protein that can integrate the signal from the insulin receptor. Next, glucose transport and metabolism. A GLUT4 gene's deletion produces mild insulin resistance and glucose intolerance. It will affect male mice more than female mice. This is because female blood glucose levels are not very high in either fasting or fat states, while male glucose levels are lower in both. It also has reduced their lifespan. However, overexpression of GLUT4 protein in diabetic mice may have beneficial effects in diabetic state, where it will regulate hyperglycemia. Other than that, overexpressed hepatic PEPCK give the result to hyperglycemia and reduce liver glycogen. Thus, it can cause GLUT4 level in skeletal muscle is decreasing, where it will disrupt the regulation of body glucose homeostasis. Thirdly, insulin secretion. It showed GLUT2, which is beta cell glucose transporter, had been reduced in transgenic mice, and glucokinase, which is sensing glucose, are the cause of the death of mice due to hyperglycemia, mild diabetes, and impact insulin secretion. Other genes like overexpression of insulin can cause hyperinsulinemia, which is glucose intolerant, as it triggers down regulation of the insulin receptor. which means it will reduce the target cell sensitivity towards the heightened hormone concentration. And now, I'm going to pass to Najwaa.
Thank you, Farah Najihah. Next, I will explain some of the benefits and issues from the usage of transgenic animals in drug development. So, some of the benefits from the usage of transgenic animals in drug development is determining the function of a gene product that results from its absence. This is by placing a specific mutation in the gene, as it may be possible with a targeting vector. Moreover, transgenic studies help to understand the control of insulin synthesis. This can be done by focusing on insulin and then process that allows beta cell to sense glucose. However, this approach might have their own limitations and issues. First, the issue that might be happen is the amount of time required to develop the animal line. This is because the timeline required to generate transgenic and knockout mice are quite long, as it requires 12 to 24 months from design to initial experimental cohort. An approach or research that requires a year or more to come to an answer might arise to a major obstruction. Next, a potentially significant reason on the issues is the use of transgenic to humanize mice. This issue needs to make an appearance, as from the recent drug development research, human protein can be used compared to animal protein, as it has a better target to identify compounds with drug potential. It is possible to disrupt the endogenous gene with a cDNA that codes for human protein. So, as for the conclusion, transgenic animals offer a valuable approach to determine the importance of a protein on a physiologic process. They may also provide to some pharmaceutical companies the opportunity to conduct a proof of principle on a target before undertaking expensive drug development work, and to use transgenic animals during the drug development process as well. So that's all from us. Thank you.